Within the Biosciences Department, we take a broad approach to pathogen biology, including fundamental discovery, focusing on the pathogen, interrogating host susceptibility and immune responses, and tracking vector-borne and disease transmission. We utilize our cutting-edge, good clinical and laboratory practice accredited laboratories and our well-established biobank platform, which has over 1 million samples that are linked to well-curated epidemiological and clinical datasets. In addition, we have over several years assessed epidemiological data and samples from a longitudinal cohort of children living in malaria endemic region here in Kilifi, and from a hospital and dispensary febrile surveillance platform. We intend to transition the malaria cohort and dispensary monitoring platform into an acute febrile cohort. Previously, though, we conducted in-depth and comprehensive analyses on the development of immunity to malaria across the whole spectrum of disease, asymptomatic, mild, and severe infections. This platform has allowed us to expand our focus into the immunological correlates in children with multiple clinical malaria episodes, a microarray platform for the prioritization of malaria vaccine candidate antigens, acquired immunity to gametocyte carriage, and exploring novel mechanisms into malaria survival, and that's using parasite-derived extracellular vesicles. The hospital surveillance platform allowed us to also investigate the mechanisms of disease using mass spectrometry, and we provided evidence that inflammation negatively impacts recovery from wasting.
Over the last 2 years, with the support from GC grant, we managed to establish a metagenomic method that relies on next generation sequencing approach that can identify pathogens in an unbiased way. So we plan to take advantage of the acute febrile illness surveillance platform to monitor the causes of acute febrile illnesses that are not associated with malaria.
We do also investigate immune factors that allow us to predict expected vaccine success in target populations. Our recent addition of the 10x genomic platform provides a detailed examination of these processes. In the lab, we have also established a metagenomic next generation sequencing method to identify pathogens in an unbiased way. And we plan to apply this method in the context of the acute febrile surveillance platform to monitor the causes of febrile illness.
We're using all this experience and expertise to study the natural history of COVID-19 infections and to particularly assess the development of natural immunity and how long it lasts in symptomatic and asymptomatic individuals. For this, we're using data from our COVID-19 testing, longitudinal immunology cohorts, as well as a collaborative study with Aga Khan University in Nairobi.
With the recent addition of the pneumococcal vaccine and the decline in malaria transmission, the proportion of admissions with suspected infection, but for which the current methods of diagnosis could not detect a pathogen, has risen. And we're uniquely placed to examine this alongside all the historical metadata we have collected over the years within the cohort.
From our recent addition to the research portfolio is the observation that chikungunya virus infections have been prevalent for a 5-year period from 2014 to 2018. Chikungunya infections have accounted for about 12% of childhood fevers in the community. Not previously reported in Africa, chikungunya-associated neurological disease was also common, accounting for about 10% of pediatric admissions with illness requiring lumbar puncture.
Given the observed increase in arboviral infections, the surveillance in the cohorts and dispensary platform will be complemented with surveillance in mosquito vectors to examine the burden of vector-borne diseases and transmission risk. The aim of this is to establish the burden of mosquito vectors in the population, to check what pathogens they are carrying, and to see if these mosquitoes are still amenable to control tools.
Furthermore, using matrix-assisted laser desorption ionization time of flight, MALDI-TOF-MS, we have developed databases for identification of East African vectors and blood meal sources, and we are currently validating these with field-caught mosquitoes. New to our research portfolio is the examination of treatments to prevent larval development of Tunga penetrans or jigger infections.
Currently, there's no information on the actual distribution and the numbers of people affected by Tungiasis in Kenya. So we have a study that's going on across the country to try and answer and get that information. At the same time, there's no registered products that are safe and effective for use in Kenya, and so for the treatment and also for prevention. So we currently have a study going on in Kwale where we're testing the effect of a neem-based product spraying on the floors to prevent the development of the larvae. And then going forward in the future, we hope to do some randomized clinical trials for the treatment of the Tungiasis fleas in people's feet.
Anti-malarial resistance monitoring continues to feature as a priority area due to the recent indigenous emergence of potential artemisinin resistance, conferring mutations in the East Africa region. For both vector and anti-malarial resisted surveillance, we continue to work with the Division of National Malaria Program within the Ministry of Health to support their activities towards malaria surveillance and control. So our expansion into the evolution of antimicrobial resistance in Kenya will provide a national overview of AMR, antimicrobial resistance, and this will include an assessment of resistance markers circulating in our community and the use of appropriate antibiotics to prevent and reduce the problem of antimicrobial resistance. We continue our work to examine genome sequence analysis of a range of respiratory viruses, for instance, influenza, endemic coronaviruses, rhinoviruses, and respiratory syncytial virus, to investigate their phylogeny and transmission from the household all the way to the continental level.
We collect several sample types, including stool, including respiratory samples. For instance, arboviruses will collect blood samples, and all that together goes into the diagnostic platform as well as the sequencing platform. Once we've done the extractions, and this being part of the molecular diagnostic process, we take them to the real-time PCR platform, where we have actually several equipment as well there, including the recently acquired Quant Studio platforms.
We've seen incredible expansion in terms of our platforms from the sort of handheld MK1B nanopore sequencing and to using the gridiron, which is excellent because it allows us to multiplex multiple samples, but also has incredible computational storage and capacity as well. And moving forward, we will be using these platforms to look at malaria surveillance in particular, as well as other important viruses of public health importance, like arboviruses, metagenomics, transcriptomics work, looking at the B-cell sequencing, for example. And that's all really, really important for our understanding and continuous surveillance of illnesses.
So working closely with our colleagues in the Health Systems and Research Ethics Department, Epidemiology and Demography Department, and the Clinical Department, we continue to apply our expertise in pathogen biology to the broader context within communities, clinical care, and health systems.